welcome everybody to this CAG webinar. My name is Pierrette Gaudreau. I'm the Vice President of CAG, and I'll be moderating today's webinar. Just a few introductory comments before we get going. First of all, as a reminder, abstracts, you can still submit an abstract to the CAG conference, which this year is in Moncton. You have today, so if you haven't started yet on an abstract, you could quickly get to it, and you can still submit it by the end of today. Secondly, just let me point out why we're having these webinars. As you may know, CAG has a number of awards that are given out each year, or some of them are given out every other year, and they are presented to the recipients at the conference. But at the conference, we don't actually hear from the recipients. So this way we have a chance to actually hear why the recipients received the award, and they can tell us about their the great work that they're doing. We're also hoping to promote the CAG Awards by featuring them again in this format, so that we get applications from other great and worthy people. Let me just note, in terms of the awards, they are posted on the CAG website. You can go there. All the requirements are listed, all the different awards are listed. And to note is that there are two new awards this year. One is for Mid-Career Award for somebody at mid-career, and a New Investigator Award. So I hope you will check those out. These are kind of exciting new opportunities for individuals at mid-career level and New Investigator level. Also, just to mention, we are muting the microphone so that there is no echo or static that is interfering. So if you have any comments, please use the chat box. If the chat box is not open on your screen, then just press on that bubble at the top right of your screen. So it's kind of a word bubble that you'll see. If you click on that, the chat box will open and you can type in your comments or question. I will be monitoring the chat box as we go along, and we will save the questions to the end, and I will then post a question to the presenters. So with that preamble, I'm delighted to introduce the speaker of this webinar. Dr. Pierrette Gaudreau is a professor in the Faculty of Medicine at the University of Montreal, and she was the 2018 recipient of the CAG Award for Contribution to Gerontology. This award, it was created to recognize an individual who has made an outstanding contribution to the field of aging. So with that, I will turn it over to Pierrette and her presentation. Pierrette, I'll turn it over to you.
Dr. Menek, first of all, I would like to thank the participants for their interest in the field of aging. And before to go through the presentation, I would like to tell you a little bit about my my training, my past training, and my research interest. So I did the PhD in pharmacology, but at the University of Sherbrooke, but I was outside extramural. I was indeed at the National Institute of Health in Bethesda, Maryland. So I touch both at my master in pharmacology and PhD, I touch different G protein coupled receptor, and therefore I was interested in that field for my postdoc. And I came back in Montreal for my postdoc at the University of Montreal, and then I was looking at there was a new receptor, the growth hormone releasing hormone receptor was just discovered and cloned, and I was interested to look at this receptor and to see if I could find some condition, physiopathological condition where it could be regulated. And there was two I would say two target possible: obesity and aging. And finally, it got a mixture of both. So this morning, the title of my presentation, From Animal Model to Older Adult, Clue to Understanding the Process of Healthy Aging. As a matter of fact, by these days, I am working both with preclinical modeling, which means animal model of healthy aging, and cohort study to look at different signature or deregulation process that could explain either healthy aging or unhealthy aging. This with the objective to find either some pharmacological agent that could contribute to healthy aging or lifestyle changes that could also help to promote healthy aging. So my presentation will be divided in 3 parts. Introduction, result from preclinical modeling, and then result from the older adult nuage cohort, nuage meaning nutrition and aging. As you know, the aging process is a very complex process which have, you know, some deleterious effect on neuroendocrine system, brain cognitive function, cardiovascular, muscle, bone, pancreatic function, for instance, insulin resistance, and fat accumulation. And like I said, I was very interested to the hypothalamo-pituitary axis because on the anterior pituitary, there was this new receptor called growth hormone releasing hormone receptor. So just a bit to see that the anterior pituitary is synthesizing and secreting many hormones, and with aging, some of them are decreased substantially. We think of gonadotropes and thyroid stimulating hormone at the bottom, but also of main interest was on growth hormone, because growth hormone has many interesting effects. Some effect on muscle, bone, and also on various processes in terms of cell production and differentiation in various tissues of the body. So we focused on this axis, knowing that about 50% of the cell in the anterior pituitary are growth hormone secreting cell or somatotroph cells. What happened with aging? With aging comes the somatopause. The somatopause is a dysfunction of the somatotroph axis leading to GHRH, growth hormone releasing hormone in the hypothalamus, so a decrease of mRNA level around 10 months of age in the normal Sprague-Dawley rat. This can lead to a decrease of sensitivity of the somatotroph to this GHRH. And what we have seen, we have reported for the first time, you know that it is some years ago, that the affinity and the number of growth hormone releasing hormone binding site in the anterior pituitary was decreased, and this process was starting around 8 months of age, meaning at the adult age. And we know that in humans, around 40, the secretion of growth hormone is starting slowly to decrease to be almost non-existent in many older adults over 80 years of age. Then we found that effectively GHRH mRNA level were really decreased. Circulating level of growth hormone and insulin-like growth factor 1 was also decreased, subsequently to this deregulation of the GHRH receptor. What are the consequences of that? An increase of fat mass, a diminution of muscle mass, and deterioration of several tissue and organs. I'm not saying here that the somatopause is the full explanation for aging. It's one of many mechanisms. Just to show you the growth hormone releasing hormone receptor located in the somatotroph cell in the anterior pituitary. So it's very similar to other G protein coupled receptor, which are a large family of protein in the body and which are the main targets for development of new drugs, also either for the peripheral organ or in the brain. So it has many specific features. This was some of the results are from us because we did extensive pharmacological characterization of this receptor. So, and we know that the highest expression is in the anterior pituitary, but now we know that there is other site that synthesize the growth hormone releasing hormone receptor. Then we will look at some results from preclinical model of successful aging. Long-term moderate caloric restriction is known for many decades as the most effective intervention to prevent or delay age-related deterioration. The caloric restriction has many positive, interesting effects that could also protect the cell from aging. Many of these effects are in contrast or in contrary to what happened during normal or the unsuccessful aging. For example, the decrease of sensitivity to insulin. So caloric restriction promote the increase of sensitivity for insulin, increase longevity, decrease the occurrence of tumor and pathologies, help to maintain muscle mass, to decrease adiposity, and to decrease oxidative stress. As you may see, there is some function here that are similar to what is doing a somatotroph axis that is working very well. So just to show you how we proceed for this type of experiment, this is a Sprague-Dawley rat that is a young adult, and this is a Sprague-Dawley rat that has been fed ad libitum for all his life. It's it's really a sedentary and obese animal because, it's like having a buffet and constantly eating up to what you can put in your stomach. And this, I apologize, this is not a young rat. This is, it could be, it looks like a young rat, but in fact, it's 20-month-old animal, but submitted to caloric restriction. We use moderate caloric restriction, which is 40% caloric restriction from ad-lib fed animals. And we install that in two sequential percentage, 20% for a couple of weeks, and then 40% up to their death or when they are sacrificed. So that's the scheme that we are seeing here. So they were fed ad libitum with casein at 8 months of age. Why do we choose 8 months? It's because the rapid growth of the animal is really past, and we are not interfering with process of development and growth. So it's like, you know, it mimics adult around 45 something age years, pardon. So because if we look at 2 months and we put them on a caloric restriction diet, you prevent, you know, the maturation of the animal, you are retarding growth, and it's not really what we want to achieve. It's really to take animal at their really mature level and then look at the caloric restriction as a model to help them age successfully. So at 8 months of age, we sacrifice the animal with 2-month-old, and we look at the hypothalamus and anterior pituitary and what it does in terms of impact on the target that we have discussed previously. So I didn't put any, you know, data in these results because I don't know the level, you know, I want to keep it simple for the audience. So if you need more information, you can send me an email at Pierrette.Gaudreau à commercial Montreal.ca, and then I could send you some more information or reprint of the papers. So long-term moderate caloric restriction, I forgot to tell you that it's a restriction of macronutrient with a balanced diet in terms of micronutrient, meaning minerals and vitamin. So we don't want to have sick animal because they are malnourished in terms of micronutrient. So what we have seen is that this type of caloric restriction prevent the deleterious effect that we have seen on the growth hormone releasing hormone receptor associated with aging by increasing the mRNA level of this protein, and then the transcriptional effect are accompanied with some good functional effect, meaning that we have better GHRH binding to its receptor, and that we can restore the production of second messenger, cyclic AMP in this case, which is crucial to stimulate growth hormone synthesis and secretion. So it's a, it suggests that with long-term caloric restriction, the receptor transcription translates to an increased number of functional receptor and reflect a maintenance of binding parameter and a somatotropic sensitivity to GHRH. Now, in order to find out what would be the mechanism of this prevention of deterioration of the somatotroph cell with aging, we look at different molecules. What we saw also in the calorie-restricted animal is that glucose and free fatty acid are increased in these animals that are at-lib fed, but they are normalized with caloric restriction. So they represent interesting candidate to study to better decipher to the mechanism of caloric restriction in mammals. So what is also interesting is that those analytes, you know, they are part of nutrition, the domain, so they could be regulated in some ways by changing dietary habits. In these animal, the calorie restricted and at-lib fed animal, we look at glucotoxicity to see if it was the culprit substance that have deleterious effect on the growth hormone releasing hormone receptor. So what we have seen is that the GHRH receptor mRNA level are regulated according to duration and severity of glucotoxicity, meaning that you have more glucose than the normal level in your blood. So in a way, 12 millimolar glucose could mimic pre-diabetic older adults that are not well controlled with medication. We have also observed that the alteration of GHRH receptor internalization is deregulate with the two highest concentration, and the 33 millimolar is a very high concentration, and if we you know, it would be a diabetic person that is really not well medicated. And finally, we could observe a decrease of growth hormone releasing hormone, stimulate cyclic AMP production with the highest level of glucose. And these were done on cell of animals. The somatotroph cell. So altogether, the cellular glucotoxicity may be one of the mechanisms of GHRH receptor dysregulation during aging. So this gives us a kind of clue to look at different aspects of the glucotoxicity in the older adult. Sorry, I don't know why, but okay, it was frozen for a few seconds. The second model that I will present to you today is the Lou rat. The Lou rat is an exceptional animal and a very intriguing animal. It was discovered in the mid-50s at the University of Louvain in Belgium. That's why we call them LOU. You know, it was in a laboratory that were interested in looking at anti-tumoral agent, and they developed you know, multiple substrain of rat, and at the one extreme they had animal that are prone to have tumor, and at the other extreme they had a successful aging rat model. So very interestingly, the lure rat, male and female, has a very long life, 90% of survivor at 20 months, while you know, the Sprague-Dawley rat that are fed at libitum are, you know, around 60 something, and with caloric restriction, see, it's similar to the lurette, male and female, and you see the Kaplan-Meier curve showing how their curve of survival and dying. So there is really a big contrast between those two strains, and it's a very interesting model because it lives a very long life. And you know, we had, this is a story, but we had one female that was called our Jeanne Calment, like the lady in France that is the oldest lady for compared to many other countries. And this female rat died at 48 months of age, which is more than the double of the age of various strain of rat that we use commonly in our laboratory. So what else can we say about the lure rat? It has many characteristics. It has, like I said, an increased longevity, an absence of fat gain, and intact cognitive function. With many tests, they act like young animals, a low level of anxiety, and an intact brain plasticity compared to other young rat from other strains. And also from ad libitum fed Sprague-Dawley rat. So it's a lean machine that we could say. So there is two targets here. So if they preserve their cognitive or function, and if they do not accumulate fat at, when they became at their adult weight, they don't gain an ounce of fat. So that's pretty amazing. So it's really a very interesting model of successful aging. The problem, and it's even better than the caloric restriction. So now we have to find out what are the mechanisms that are responsible for these really impressive longevity in good health. So we up to a few years ago, it was not, the genome was not sequenced for the lure rat. We did that, and we are in the process with collaborator in Paris, a genetician collaborator in Paris, looking at the sequence of the genome, comparing this strain with many other strains of rat, and apparently there are many differences. So it's gonna be a long way, but at the end we could have some very interesting target to look what happened in human with these protein, these different protein that or locus of the genome that allow for this exceptional longevity. I have also other result with my postdoctoral student at the time, Dr. Caroline Ménard. She was interested in memory, hippocampus memory, and we said, well, we will challenge them for a long period of time with a very high fat, high glucose diet, and probably either they will not be able to cope with that, and we will see a lot of genes that will be dysregulated, or they will be able to cope, and then they will develop some mechanism, and we should see one way or the other, we should see some changes in gene transcription. To our disappointment, we didn't see, and we had young and old Lou rat that were submitted for 4 months to this regimen, and any young rat from other common strain, they become insulin resistant and obese with this type of treatment. And it did nothing at the level of the hippocampus, no regulation of any gene. But we look also at the hypothalamus, and we see some regulation, small regulation that were not preserved at the level of protein, but some regulation at the mRNA level for one protein in the leptin axis. The leptin axis is, the leptin is one protein that is secreted by fat cell, mainly fat cell, and tells to the hypothalamus to stop eating. So we saw some little deregulation at this level. And what was also very interesting and intriguing was the fact that they had like an internal sensor that prevent them to eat more calorie than their pair animal that were eating the regular diet. So they have some type of sensing in the brain, I guess, maybe also in the periphery, that prevent them for eating too much and gaining weight. So it's a very interesting model for either discovering new molecule that could be related to obesity and also to memory. So we are working very hard on that. Now I will turn off on the cohort study because these information that we gain from our animal model should help us to question, to formulate some new question in research for the older adult, to their benefit, because that's the ultimate goal of our research, to be able to do something to assess that the older adult will have a longer life in good health, and stay at home as well, be able to stay at home. So in 2003, we were fortunate enough to have a grant from CIHR, and we launched the longitudinal study Nuage for nutrition and aging. So, and the focus was to look at the determinant of successful aging on the side of the nutrition. So it was an interdisciplinary and plurimethodological research program, and the big PI was Dr. Hélène Payette, who is now retired, Dr. Gredonald, who is also retired, Dr. Schattenstein, who just retired, and myself and my colleague, Dr. José Morin. We are there to pursue, and we have new people with us to continue this adventure with the Nuage cohort. So, like I said, the conceptual framework was nutrition, and so looking at the past history in terms of health, weight, food, and socio-demographic information, what was also that was also have some effect on food intake, energy and nutrient intake, anthropometry indices, and biochemical indices. Altogether, this will lead to different condition in terms of physical activity, endurance and strength, body composition, oxidative stress, and so on. And the main outcome will be to maintain functional capacity, physical and mental health status, and social functioning. So it's a very big study in terms of quantity and quality of data that were collected. So, like I said, this was a pluridisciplinary approaches, including basic clinical, epidemiological, and social research. We recruited roughly 1,800 men and women, divided in 300 age group for men and women. So we had people from 68 to 82 in 2003-04. So we also set up a large data bank for, saliva, urine, serum, plasma, lymphocyte. As a matter of fact, we are actually waiting for some exciting result, because we will have soon the just all the single nucleotide polymorphism of this cohort, and so it will be very interesting to associate that with some trajectory of these participants. So we look at different facets of nutrition, at functional, medical and social aspect of these people, and the data were collected by questionnaire and a lot of direct measures. Many sub-studies were conducted using these data. As an example, the NUT-COG, Nutrition Cognition Study, who look at the impact of the nutrition on cognitive function. What we have done is 10 to 12 years after, which was in 2014 to 16, we were able to get some new money to have a telephone interview with these participants. And if you calculate rapidly, so we had a good number of non-Nigerian in this subgroup. Very briefly, the cohort was a random sample stratified by age and sex, obtained from the Quebec Universal Health Insurance Plan. The criteria of inclusion were the following. It had to be community dwelling men and women, age 68 to 82, French or English speaking, willing to commit for a 5-year period, free of disability, inactivity of daily living, no cognitive impairment, able to walk without help. A cane was okay, able to walk 100 meters and climb 10 stairs without rest, able to sign an informed consent. So what we see here is what we have done in the first wave, and then with some additional money from the Fonds de Recherche du Québec Santé, we were able to do the telephone interview. The criteria of exclusion were, heart failure problem, pulmonary problem, inflammation, digestive diseases, and cancer. Of course, all those influence nutrition, and it was, we really want to have some community dwelling people that were in general good health. Okay, so briefly, this is what was computed. We had almost 1,000 variable per year per participant, and we had also a lot of sample, around 55 to 60 different sample of various biofluid that we collected yearly. So people came to research center for older person, both in Sherbrooke and Montreal area in the Quebec province. And so they were almost one full day at the research centers to have these many measure and questionnaires on their various function and habits. In 2014-16, what we did was the following. I remind you that it was a telephone interview. We tried to ask these people to come to the research center, but like I said, they are quite old, so they were not very interested. However, for some other project, sub-studies, they had some people going to their homes, and they were really receptive to this idea. So for future, it's very interesting that we can seek for new data from these people if we go at home. So we did two questionnaire for cognition, and we did 3 other questionnaire for, malnutrition, perceived functional limitation, autonomy in the activity of daily living. What did we get? So we had in 14-16, we had 747 person that we could reach. That's pretty amazing considering their age, and 33% of the initial group accepted to answer the various questionnaire. Depending of the person, it could take 20 to 75 minutes. So we had almost 300 men and also women, so a little bit less of men. For this questionnaire on memory, which is rather difficult, we had a little bit less person because it was started with some delays, and so we have a smaller number that of data that were collected. The age range in 2016 was 80 to 95 years old. So this means that actually we have a good number of Nigerian in the cohort. Okay, what they look like. So if we look at At the male, we had, like we said, a little bit more women than men, and most of them were still married. So a smaller number were widowers. What were they when they were working? A good number of men and women were professional. Others were in, administration and commerce or merchant. Some women were technician, and many men were, labor working force. But we had also a good deal of women that were women at home taking care of the house and the children. So what they look like after all this time. So if we look at the body weight, we had really a tremendous window, and it's the same for women. So it means that we had really a good number of persons that were more than they had a little bit of overweight, if I can say so. When we look, we ask them to see if they lose weight over the last year. A good number of them said yes. When we asked them if they had a good appetite, we had about the same number of people saying yes. And we did not have a lot of people. When we look a little bit further, we don't have a lot of people that have really no appetite, or really few of them. When we look at the nutrition screening tool for malnutrition, we see that it's not the case for these men and women. When we look at the MiniMental, when we look at the perceived functional limitation, for example, are you able to raise your hand above your shoulder? We see that a lot of them had no difficulties, both men and women, and also they had a very good score in the autonomy of activity of daily living. So they don't have a lot of, they perceive that they don't have a lot of limitation, and they are able to do what they want to do in the activity of daily living. However, when it comes to cognition, and it was another story. And if we look at their score, we have really a big window there, meaning that we have people that are not, if we look at the lower limit, considered normal according to the education level, we have a good number of persons that are under this level. And when we look at with the other cognitive questionnaire, which is more difficult, we had even much lower, much lower result. We had poor score. It was judged long and difficult for the nuage participant. And we rely on a publication that was saying that it was taking a little bit less than 10 minutes and was not difficult. Well, it was not the case in our nuage cohort. So what does it tell us? I think if we look back at our successful aging animal model, we have many elements that have similarities, like what is the impact, for instance, of the body mass index status. How is the fat interfering with the memory of these older adults? How many of them are pre-diabetic? And that could interfere also on their memory. So actually, we are looking at the trajectory of these people, looking at diet, exercise, social participation, chronic disease, link between diabetes and memory. And we are seeking for other NUASH participants that could be recontacted. But the good news is that we should have a big grant soon that would allow us to go at home and make more of these really survivors. So, and then this will help us to, you know, to have new data on, you know, analytes, serum analytes. Also, we will be able to have a detailed questionnaire on food consumption and so on, and take a measure for strength and look at their memory with different questionnaire as well. Secondary, we will be able to look at some modulator with change in gene polymorphism, example for ApoE44, which is a risk factor for Alzheimer's disease. And we will be able to question to question whether or not lifestyle, mainly diet, exercise, social participation, influence positively and significantly healthy aging and the relationship, and to see if the relationship could be modulated by genotypic variation. And finally, what we are doing actually, which is another project very exciting, is to look at some participant of our cohort to define a metabolomic signature of metabolic syndrome. And this is an ongoing international study with a researcher of the French Metabo Hub. If we are actually writing the paper on that, and this could help to stratify people for more individualized treatment when needed, and to give better assurance that we have a signature that permits to have more a better grasp on the unsuccessful aging, and having more meaningful lifestyle intervention or drug intervention to keep older adults in a better health mood. So this is about what I wanted to say. This is a really very macroscopic overview of what we are doing in the lab, but I should say in collaboration with many researchers in Quebec. And I would like to acknowledge the participant and NUASH team, because it's now a very big study. And like I said, we will go back to see these persons. We are, are actually filling the paper at the government to send to the government to have access to their clinical data and the age at which they die and also from which chronic disease. So it will give us really a full spectrum of the aging of these people, and we will have for a good number of years to ask complex questions to better identify determinant of healthy aging. I would like to acknowledge the work of Karine Bédard and Caroline Ménard that were students in my laboratory, and also the CIHR and Fonds de Recherche du Québec granting agencies that allowed to do these very interesting, I would say, passionate work, because it takes passion in research to succeed, and it takes it takes also a lot of dedication, but it's good because we are doing that for our older adults across Canada. Finally, I just want to thank the Canadian Association of Gerontology for the prize I got last October, and I am a real supporter of this organization that allowed to gather the research in aging community across Canada. And I thank you for your attention. I will gladly take some questions if you have.
Thanks a lot, Pierrette, for this very interesting presentation. And you know, I never thought I would say this, but I would like to be a loo rat. That sounds like a
Me too.
pretty, wouldn't we all, eh?
Yes.
We're 2 minutes before the hour, and so we just have a little bit of time. I would actually like to ask you a question before we close this webinar around the Canadian longitudinal study on aging, and which was in many ways modeled on Nuage. So there's clearly a similarity there in terms of the measures that were in the studies. What do you think are some of the opportunities within CLSA to take those animal models that you've been using, but putting them into a cohort study? Any thoughts on that?
Well, it's complicated because
I'm putting you on the spot. I know.
Yes. I think the finding that we can find in loo rat could generate some interesting questions, both in our Nuage cohort and also in the CLSA cohort, that's for sure. But what I am more looking for is, since we will have we should have some new data in 2020 with the people that we will visit in the Nuage cohort, and then we can, look at these data and correlate some data that we have on loo rat and Sprague-Dawley rat, and then with this information, generate questions to look into CLSA, because it's a larger cohort. So in a way, it could serve. I would say that Nuage served to validate some question or some result that we have from preclinical modeling, and then CLSA will serve to Nuage to validate our hypothesis on a larger cohort.
Yes. Okay.
Is it the type of reply that you were looking for, or am I am
Well, I
correct?
wasn't looking for anything specific, but yes, thanks a lot. That makes a lot of sense to use Nuage because it has a lot of nutrition questions, more so than CLSA, that I can then lead to questions for CLSA as well.
And
anyway, sorry.
Yes, I was saying that in addition, in Nuage, actually, we have a lot of data on serum biomarkers that are not necessarily ready now in the CLSA. So that's why I am, it would be a continuum for translational research from our animal model to Nuage to CLSA.
Mm-hmm. Okay, with that, and we're just past the hour, so I would like to close this webinar. Thanks everybody for attending. Again, remember that abstracts can still be submitted today if you work really fast. Also to check out the awards on the website, and I wish you all a good afternoon. And thank you, Pierrette, for this presentation.
It was my pleasure.